people generally don't relate to plants as much as they do animals, right? And I think that's just because we are animals ourselves.
Yeah.
Plants being taken out of the wild, or wild harvested is another term you could use, does significant damage to those populations, especially when there's a high demand for plants, whether it's compounds that are very important for us or it's food resources.
Hello everyone, good evening. Welcome to First Fridays. I'm Joel Keyson, Program Manager here at NHM. There will be no animal genitalia talk or images tonight, sorry. But we're talking about plants. Exciting, So our theme this season is From Feelings to Pheromones. We hope to explore the forces that shape the behaviors of life forms large and small, from reproduction and propagation in the natural world to the sex life of other species. We'll untangle the constructs of gender, feelings, and pheromones. And now, without further ado, on behalf of NHM, it is my pleasure to introduce our host and moderator for First Fridays this season, driven by a mission to redefine where science storytelling belongs and a belief in the power of science to connect people and communities. We are truly fortunate to have her as our moderator for another season. So please welcome back neuroscientist and science communicator, Dr. Yewande Pierce.
Thank you, Joel. Hi, everyone. It's so good to see you all. I am super excited for the discussion tonight. As my husband will tell you, I'm a huge plant lover, so I've been looking forward to this one. So welcome to this evening's discussion, the Micropropagation Connection, our relationship with plants and how cloning plants can affect the greater biosphere. So tonight we're going to be diving deep into the world of Catalina Island's rarest plants with our expert, Rare Plant Ecologist, Kevin Alison. We'll be exploring the cutting-edge applications of micropropagation in fields like agriculture, horticulture, forestry, and plant breeding. And we'll be discussing why cloning plants has become a rapidly growing trend. How does micropropagation influence the plants in our everyday lives? What implications does tissue culture hold for us? And importantly, how can micropropagation contribute to preserving endangered plant species? These are just some of the questions that we're going to get into tonight. So without further ado, I'm excited to introduce our guests for this discussion. Please welcome Kevin Alison.
Hello, people.
Kevin is the Rare Plant Ecologist for the Catalina Island Conservancy. He uses his education and experience as a conservationist, plant scientist, and horticulturalist, lots of ists in this intro, to ensure the rare plants of Santa Catalina Island do not go extinct. So no pressure there. Thank you for joining us, Kevin.
Oh, my pleasure.
So to get our discussion tonight started, I wanted to just delve back into your background a bit. You have been really fortunate to live in some of the most beautiful natural landscapes, including Laguna Beach, Palm Springs, Hawaii, and also Catalina Island. So how have these locations shaped your journey with plants, especially the rare plant species that you have become familiar with?
Yeah, so of those really beautiful places here in Southern California, a few of those that you mentioned, including Orange County and down in Laguna and here in LA, Palm Springs area, Hawaii, and Catalina, all those are biodiverse hotspots, really. And even areas here in California, Southern California and into the deserts, people consider maybe really dry climates and maybe not a lot of plants grow there, but there are just a lot of plants that have adapted very well to our climate and these harsher, drier climates in that way. And there's an immense amount of plants in these regions. And so all the way from here on the coastal climates and how the land meets the sea and how that interaction occurs and the different ways that the plants have interacted with that type of environment, all the way out to Palm Springs in the desert where they've had to adapt. And we have some of the driest deserts out in Palm Springs in the Colorado subsection of the desert. there's an incredible amount of biodiversity in those, and plants that had to evolve very special traits to be able to do that. And then I went from Palm Springs all the way to Hilo, Hawaii, which was like one of the wettest places, and
quite a
just had a contrast. But it was interesting because it was a way to see plants, especially in Hawaii, where brand new earth is being made, with the lava flows and everything like that, and seeing the succession of plants and the adaptations to that type of environment as well. I saw this whole gamut, the diversity of how plants evolve through many different climates, basically around the world in some ways. I didn't hit Antarctica or any of that, but
Not yet, but
just the cooler places like California and Hawaii, but no big deal. But and yeah, so that's where I really got to learn a lot of how plants work and how they survive here on the planet.
Thanks for sharing that. I think what's really interesting about where you have come to specialize is this intersection of horticulture and conservation. All these plants are around us all the time. I don't know if we'd all kind of be able to spot the rare species or be immediately invested in them. But would you say, as time has passed, you've become obviously more passionate about these rare plants, or did this start early on in your childhood, and what actually got you into it?
When I was young, I used to always go out and make forts out into the wild, like a lot of people, I hope, and really get to experience the wild. And certain things were rare, but I may have not known that, but there was always opportunity for discovery. And being like, well, what is this? And then I think in today, when people have, AI on their phone and they can just snap a photo and know what something is, it's pretty cool. But for me, I was always interested in the outdoors, but didn't even know until I got a little bit older that I came from actually a line of horticulturalists. And it kind of skipped a generation, but when I got to know my grandmother, kind of in her later years, she taught me horticulture right at the perfect time when I was wondering what I'm going to do with my life. And as soon as it was like, oh, this is it. And she was, she started to lose, she could barely walk, but she was so stubborn and would refuse a knee surgery, and she would walk around with a cane, and she was this really interesting, goofy woman. I loved her to death. But she taught me how to take care of her garden in her backyard. And she was so enthusiastic about every different species and everything like that, and these rare collection plants that she had. And it was so contagious. And I was like, wow, okay, I know what I wanted to do. And I got really interested in growing plants as well. So, and methods we call propagation, or being able to create new plants by seeds or cuttings or anything like that. But I was always interested in the wild and going out on hikes. And when as a kid going out into nature, you're like, well, I feel like I know that tree is my neighbor, right? Like I know that that's Jeff. Sorry, Jeff.
Name the
but but to feel that way. So when you start to really know some of the plants, like Laguna, we had one succulent that grows in the canyon where I lived, and I had no idea that it was exceptionally rare, occurred only there, and was from there. And that plant, just like me, were from Laguna. So it became this really relationship where I'm like, we can start to build and use horticulture to help save a lot of these plants as well. And so that's where I kind of understood my career path is going towards using that passion towards just growing plants and appreciating them in that capacity, and getting to know them pretty well by doing that. As well as appreciating them in the wild, and how can we continue that for future generations. Yeah,
and I think ending up in Catalina Island, where there are so many rare species that we'll get to, and having that background and that informing your investment in this work, but then also, as we'll see, the micropropagation is super interesting, so I'm excited to delve into that, but developing these techniques that could actually help us preserve these rare species. It's really cool. So plant poaching is the next thing I wanted to talk about, which is it's something that I don't think everyone is familiar with. So I wanted to spend a little bit of time talking about it. So Catalina Island, it's home to over 700 native plant species, if I'm correct, with several found actually just exclusively in Catalina Island. So it's really unique to the island. We've heard about habitat loss because of, building and also natural disasters like wildfires having an impact on those plants. But plant poaching, I think, is something that's beginning to emerge as a significant threat to the natural flora. So it'd be great if we could spend a bit of time just talking about what plant poaching is and why it's so detrimental to these rare species compared to the things that we're more familiar with.
Yeah, so poaching is exactly that. We think of people may have heard, I mean, we're surrounded by animals, a weird audience included in here. I know, but we've typically heard about poaching like rhino horns and, or even the animals in general from the wild. Some big cats for fur and things like that too, but plants have also had a lot of poaching that doesn't necessarily get a lot of the attention that I think people were aware of. And so, no offense to plants, which I would take offense to plants if people talking like this, but people generally don't relate to plants as much as they do animals, right? And I think that's just because we are animals ourselves.
Yeah.
But plants being taken out of the wild, or wild harvested is another term you could use, does significant damage to those populations, especially when there's a high demand for plants, whether it's compounds that are very important for us or it's food resources. That's when people really have awakened to sustainability and those types of farming and cultural practices. But there's been a new trend that a lot of the cactus world has really dealt with, the carnivorous plant world has really dealt with.
Succulent, yeah.
But succulents as well, and this one particular succulent, which we'll get to, here, and that's local to only the coasts of California, scrapes a little bit into Oregon, but all the way down into Baja California as well, really kind of awoken us to an awareness that plants were being poached here locally in great numbers. So I think we have a slide to show.
Yeah.
In about 2018, maybe a little bit earlier, I forget the exact date, but when I was looking towards my grad school work, I went to Hawaii to learn the micropropagation technique, which is just a more advanced method of growing plants.
Yeah.
And then I started a career working at a native plant nursery with about 40 acres where we grew a lot of native plants. I was looking towards what do I want to do with my grad school work? Because I was really interested in doing micropropagation. Yeah. And then all of a sudden, this hit the news with my favorite genus, which actually was one of the that succulent I mentioned in Laguna Beach, where there's a rare species that occurs there, was being poached up in Northern California. This is Dudleya farinosa. I forget the common name. Sorry, I'm a plant scientist. I don't remember specifically. Yeah, I'll speak some Latin, but I'll speak it very softly so it sounds very nice. Dudleya farinosa. Yeah, but it was being taken out of the wild by the tens of thousands in some cases. This is just one event.
Wow.
This was a separate event over here, but this photo is cropped. It goes like much further down as well. And they actually found these people off of a tip. They thought they were poaching abalone off the coast, but really they were scaling the bluffs and collecting these succulents.
Wow.
And so they pulled them over, and then they traced them back to where they were staying and found much more, and being shipped over across to supply markets overseas. So this plant is called commonly the genus is commonly called live forevers. And so some people have speculated maybe that's because of,
medicinal properties.
Yeah, something like that. But really it was about the succulent craze.
Wow.
That succulents became really popular. They knew that they were able to grow. But this dudleya that occurs in California, the Californias, I include Baja in that, have this really unique rosette shape. They have a chalk that no other real plants really have, and it became very popular. But poaching them out of the wild became a really popular thing. And amongst really collectors, the rarity of some of these were even more desirable. right? These are like, I could get my hands on something really unique, and that's a huge problem in the wild when there's entire populations being taken out.
I mean, that's well, because you said hundreds of thousands. Tens of thousands.
I mean, we don't know necessarily.
You don't know. It's hard to quantify. And just going back to your point about how these rare species live amongst us, and you're already passionate about plants, but you don't know if they're rare. To think this just happens, like just under your nose, and is going out globally to markets is really shocking.
Correct.
And so, I mean, poaching is clearly going to impact the local environment, but I'm also curious as to how it impacts where they're going, if it's going directly to market, and whether, for example, plant lovers like myself would be able to know that it's being poached. Is this happening without us actually being complicit? Or
so us and the authorities were wondering the exact same thing. This was relatively new to us. We knew a lot of this was happening in some of the areas where cactus and other rare ones out in the deserts, but we didn't know this was happening on the coast. And really, these plants were going to, I think when they started to trace it back, was to areas primarily Korea and China. But I think it was mainly in Korea, as kind of, there's some exceptional horticulturists out in Korea. And I think there was kind of the central hub that would then supply a lot of the markets overseas.
I see.
And so it's just, it comes down to just different values, different ethics, and those types of things where, you know, and whatever it might be, the supply and the economic aspects of how much these were being desired for was the driving factor. And so when with my grad school work was like, okay, we can do something here using the principles of economics by increasing the supply of these rare plants. We can flood the market and decrease that demand. And so using this principle of supply and demand with economics, we can actually do conservation work through horticulture.
I love that approach, that classic demand and supply applied in this setting. And I mean, there has to be an incentive. In terms of the markets that these plants then ending up in, is it just in terms of like, you know, for display, or do they actually end up in the ground sometimes? Because that creates different problems in terms of...
Yeah, a lot of people like that kind of wild look to some of them. Nature does an incredible way of sculpting them, because a lot of these, as the term for this plant, live forever.
Yeah.
A lot of these are very old specimens. So some of them could have taken decades and decades to get to a really mature shape and unique structure that looks so natural. And so this has happened even on Catalina, where we've caught people just taking plants, and they would grab the biggest ones and probably the oldest ones too, that have just been shaped beautifully and structurally over time by the winds and all the different types of elemental things that have happened to those plants. So wild plants were kind of desired for in some cases, but really it was just about accessibility. It's just they wanted these species because there's nowhere else in the world they can get them, and it was new to the markets, and it was new to collectors and that wanted to collect, you know, plants and have them in their garden. And so unfortunately the supply was so low that it was worth them going across the world to wild harvest them and ship them back. And so that's the problem we were trying to tackle, is let's make this so ubiquitous. I hope you buy these and accidentally kill
really
them. No offense. Buy them at Home Depot for nothing. And so it was a hard sell to my boss when I was like, let's grow these in mass and sell them for nothing and make no money and everyone will be happy.
Like, come again.
Yeah, but when I told them about the ethics and like how important this was, and we were in a unique position to do something about it, having a micropropagation lab, they definitely came around with that. So.
Well, I'm excited to get into micropropagation as a conservation tool because this is pretty new. Like over the past decades, as we've touched on, succulents especially have become immensely, immensely popular. And you advocate for micropropagation as a critical tool to conserve rare species and safeguard against not just poaching, but also overharvesting and talking about that demand and supply issue. So it'd be great now if we could just break down the process and talk about how it offers advantages over more traditional conservation strategies like seed banking, for example. I think next slide we have a good visual.
Yeah. So before we get into the sciencey, stuff and diagrams, but so first we'll define what traditional methods are.
Yeah.
So traditionally, when we grow plants, and we've been doing this for eons as people, is we've been collecting seeds, storing seeds, holding, and we're really precious, about seeds, and then being able to sow those seeds and then have whatever plant it might be and whatever products that we get from those plants. And that's how nature has evolved to do, a lot of continuing of generations of plants. And some plants have learned in strategies of actually doing asexual reproduction or cloning in some way, but like a strawberry plant has a runner that goes out and then a new plant forms. Well, that's the exact same plant. They're all connected, but it could be divided off, and with erosion or something, that plant can then survive on its own. But it's actually, then you have two different individuals, but those are actually identically clones of each other.
And that's genetically identical.
Yeah. Cactus pads, they also will fall off and sprout roots and then grow. As many people have seen cactus all on the cliffs and things around here in the coastal California. So those are all strategies that these plants have used asexually or clonally to be able to grow themselves. And so we've done that for food crops and all different methods. But what happens, especially with rare plants, and we want to conserve them, what do you do when those traditional methods don't work?
Yeah.
So either the populations are so low that they're just not producing seed, and we'll talk about some species on Catalina that are doing that. Or what happens when, there's a problem with seeds as well, so let's take a cutting of it, but that doesn't work. It's so impossible. It's usually a hardwood plant that doesn't want it. It just is so difficult to root by cutting and then have a new individual. And that's where micropropagation gives a way to alleviate and overcome those challenges. So how you do that is you grow plants in a test tube for the most part. So it's taking a cutting or even a leaf tissue in some cases, and you're able to clone that plant in a lab by giving it a nutrient gel solution. So if you can see up here, in the first stage, you take a little cutting, in this case, the little W graphic.
And this is just any cutting from the plant of interest.
Typically it's any growing point.
Okay.
So if you can see on this side, this is a rare manzanita, Arctostaphylos. And typically we just take cuttings of those, right? And you can do the same thing, but we stick it in a nutrient gel solution. But first you have to clean it, because this has all that nutrients and sugars and all kinds of things in that gel that bacteria and fungus just will thrive and take over this whole jar. So we have to aseptically or clean these plants completely and then have the perfect conditions for these plants to grow with no competition with bacterial or microbial type of contamination. They have all the light they need. There's no shortage of nutrients, and a lot of times we have to figure out what nutrients they actually want or don't want. But we can do that and control that within this environment. So the temperature, everything like that is able for them to grow. So once we have them nice and established in the laboratory setting, then we're kind of in the driver's seat of what we want this plant to do. So then we want to make more plants. So we're going to actually use either synthetic forms of natural plant signals or hormones, because that's really all hormones are. They're actually just signals on what cells
them
want to do.
grow.
Right? And then that's all based on the concentration of how much and what kind of chemical or what kind of hormone. And we can do that. If we want the plant to produce shoots, in the case of multiplication, that's perfect. Now we can have new shoots and new individual plants or new cutting of opportunities to emerge. If we want roots to form, we can just put a rooting hormone in there, and then roots will start to form. But there's really two methods to be able to replicate plants. So in the case of the Dudleya, that's a small rosette, you can't really take a cutting off of a cutting like a normal branching tree would. So in that case, I had to go to what's called callus tissue.
This bottom left image
bottom left. And so in this case, you actually induce to make a cell mass that's essentially like a plant stem cell. They're de-differentiated cells. It's no longer a leaf tissue. It's no longer a stem tissue.
It's like step one.
It's just a plant tissue. And then from that, we can then use those signaling hormones to say, okay, now I want you to produce a shoot. And then that will then be like, okay, so then every cell has the potential to now be a new plant. And so a new clonal individual, if you use one source plant, and it worked actually way too well.
This is incredible. Yeah.
So from
amazing.
one plant, I was able to then, there's hundreds of plants
That's
there.
beautiful.
And this is a rare plant that only occurs on Santa Rosa Island. It's called the Munchkin Dudleya.
A Munchkin Dudleya.
A Munchkin Dudleya. It's only about this, it gets about that big, and it's, but highly desirable in the horticultural trade. So once you have that, new plant form, you can go to the next slide. Then we, if you look in the top left here, then we acclimatize them. So now they're ready to go. If you root them, we were lucky with Dudleya that you didn't need to root them in vitro or inside the gel or inside that container. They would do it sometimes, but great. But you could just transplant them carefully or acclimatize them or create a new, or get them ready for their new climate out into the greenhouse. So if anybody's grown plants before, sometimes cuttings, things, you need to get them ready out into the wild.
Yeah.
You do the same here. So this is Lillian.
We have some actually at home.
Yeah?
They're not ready yet.
Okay. So this is Lillian. She's taken over the lab work and all the micropropagation of the Dudleya since I've left and now on Catalina. But here she is bringing those plants now out. It's a really rare one, actually from LA area, Veridii, and getting them ready to root, get it ready into a garden or somebody's garden, or eventually maybe to somewhere like the Natural History Museum that uses it as kind of for education and all kinds of other places. But we know that we've ensured that more of those plants have been grown and that species will no longer go extinct.
Because it's so multifaceted and so many levels. So one, your idea of flooding the market, that poaching incentive removed when you have a ton of them. You're also preserving them instead of seed banking, you have a backup plan. But then also restoration because they're being put back into the areas where perhaps they've been poached. But I do have a ton of questions. So from the previous slide, I imagine, so you were talking about the cocktail of hormones and you can almost just drive their identities. Does it have to be specialized for each rare plant? So you have to do this process each time depending on the plant that you're trying to culture, or are these more ubiquitous mechanisms that are at play?
You nailed it on exactly the biggest challenge, is that these plants are, especially when in the case of something rare like this has never been done before.
Yeah.
And so it was a whole new endeavor for me to be like, okay, where do I even start?
Right. Which hormones?
Which hormone? What concentration?
Yeah.
And or even just getting it clean into the in vitro. Okay, what concentration of the bleach solution? And how long do I expose it to that bleach solution to kill off all the bacterial and fungal and microbials without killing the plant?
Because plants are part of an ecosystem and environment, so I imagine that there are some things that they do rely on to thrive, perhaps.
Exactly.
Maybe not bacteria. But
So a lot of unknowns, and so that's where science comes in. So we start to use different methods, try different concentrations, and monitor, and that will drive us to then which ones would be the right, appropriate thing for this plant.
Well, that's great. I mean, as an ex-cell biologist, you've explained it, you've explained micropropagation as being tissue culture, or you could call it in vitro propagation, which means in a test tube. And so with my background in tissue culture, I immediately think of challenges, that some of which you've mentioned, like contamination, we touched on costs, genetic stability, but then also scaling up. So how you actually scale up is a huge question mark for me. In a lab, you can do this, but you can also do it DIY style. So what is the difference between trying to do this in a lab versus if, someone in the audience wanted to try this technique?
Yeah, and we're at the point now with this technology of micropropagation where it is available to the home gardener. And there's plenty of YouTube videos and other things like that where if you wanted to try this method, you can at home. But for a while, it was kind of restricted just to university researchers that were able to get access to certain laboratory equipment and nutrient gel solutions and all that type of agar and all this type of stuff, and the tools to even do the sterilization and a laminar flow hood and all this type of stuff. But it's the challenges are now it's more available, but it really goes down to the science, and so being able to scale up is okay. What is the most efficient way to do this, and what works pretty well or very well that makes sense to then to scale it up to larger models and make that more cost effective to be able to do that initiative.
If we could go to the next slide, please.
Oh, one back.
One back.
Yeah.
Oh yeah, we're on this one. So in your experience with all of these rare plants, there are common roadblocks that you find. Is there sort of one plant or rare species that has worked really well? You mentioned the Dudleya that we have, and that was beautiful to think that that was a clone. But kind of in terms of scale, is this something you could do with most rare species, or is it that you've nailed like a handful and there's still quite a way to go?
In my world, it's so I'm trying to do a lot of propagation of really rare plants, and this is kind of new, especially for Catalina, where we're using these this method to overcome some horticultural challenges. So we're in the new phases of that. There's a lot of examples of other rare plants that has been successful for. I just know just recently the Santa Cruz Island, which I'm going to go in a couple of days here to go see it. They were able to finally get this really rare tree called a Berberis pinnata insularis. Sorry, I don't know the common name for it, but there's only about 12 small pockets where this plant occurs now on this island, and same situation. They weren't able to get seeds. Cuttings were really difficult, and they used this method. I think it was at the Huntington Gardens. They were able to finally, after years of research and they were able to finally get this plant able to be well micropropagated and back. They had a lot of challenges with that species, just not behaving the way it wanted to, but they wanted to. But hey, we're doing a weird thing with this plant, and we're the same thing with our rare plants on Catalina. So this one here is actually the rarest tree in all of North America.
That's the top right.
Yeah, this is the Catalina Island Mountain Mahogany. And I say North America because Hawaii has us beat on that, but they have exceptionally rare species and everything. But this one, there's only, as far as we know, 6 pure individuals left in existence.
6?
6 plants.
Wow.
And these are older, mature trees. It's in a rough part of the island, and it's in one central location. And if we have one fire, a devastating fire or anything like that, or a flood, or who knows what, disease, new pest or something comes through, we could easily lose these plants. And these plants are getting old. And unfortunately, they're not making any more seed. So
I was gonna ask
on this slide here, they're making seeds.
Yeah.
But what they're making are hybrid seeds. So it's hybridizing with a very common plant that occurs throughout the islands, on the other islands, and actually here on the mainland. very similar. It's hard to tell the difference, but it's actually hybridizing with them, all the pollen pressure on those plants, and now any seed that comes up are now no longer that plant. So that plant is basically getting bred out of existence. And human-caused things, we had a history of ungulates, and still do, on Catalina, that have caused the rare population of plants that had less adaptation to these browsing animals that we brought to the island. That population got so small, now you have a common species that we call genetic swamping or hybrid swamping, making that seed it's just having a harder and harder time surviving and existing on our planet. And so we basically lost seed capability, and any seed we want to use for restoration is no longer really that plant.
Available.
And so that's where micropropagation is.
Key micropropagation.
And so if you see on this bottom here, it's just another plant. Okay, we lost the ability to make seed, and cuttings are like almost impossible to do. And so we just don't have a reliable method of ensuring that these plants go have a future into the future and have new propagules until we can figure out a way to clone this plant. And so that's where the micropropagation comes in. And then when we clone it, we're actually capturing all the genetics that are there.
Preserving it. So it really does maintain its integrity. You don't have that issue of hybridization. And then when we were just chatting before the discussion, you mentioned something pretty cool about how you can take it a step further and introduce the idea of cryoprotection, which is being used in the animal world, and thinking about conservation. How would that sort of feed into micropropagation? Sort of how far along are you with that?
Micropropagation is the intermediate step for plants to get into in vitro culture. So seeds, you can do cold storage of seeds, and in that is a little embryo, but it's really difficult to get those to survive cryopreservation. It gets a little too cold, too dry, and things like that. So, but we can actually create somatic embryos or embryos from somatic cells, which means asexually. So these are basically a baby plant that created an embryo without sexual reproduction.
Completely asexual.
And it's completely identical clone genetically. And so plants figured this out.
They're ahead of us.
Pretty well. Whether those are able to survive in the wild, I'm not exactly sure, but we're able to do it in the lab. It was kind of when you saw that mass of cells, of that callus, you'll see little heart-shaped little forming of cells, and that's actually when they discovered, those are embryos.
Oh, wow.
And that we can actually use, and does well through different techniques, to be able to put them into cryopreservation, or really low temperatures with liquid nitrogen and other things, to be able to essentially preserve these indefinitely. So I was actually there this morning, just coincidentally, for a conference. Down at the San Diego Zoo, they do cryopreservation. The Huntington does it. I think the Lion Arboretum out in Hawaii might be doing it. Morton Botanic, a few places are doing it, but a lot of them are doing it with, especially at the San Diego Zoo, with animals, but they want to get plants in there too. So if you go to the next slide, this is an ironwood that occurs only on Catalina, and it's actually a relic species. It used to be widespread throughout the whole western region. I think almost out to Death Valley or out to Utah or something. And over time, as climates have changed, it occurs now only on the Channel Islands, and this particular one only occurs on Catalina.
Okay.
And it's one that we have 120 groves around the island. We found out that each one of those groves are actually clones of each other. And so all those trees...
Wow, just naturally
So we actually only have 120 plants, where we thought 120 groves with all these trees, we have thousands of plants. No, actually we only have 120 individual plants, really. And they're not producing seeds. It's another one that's like really impossible to take cuttings of, and that's where micropropagation comes in. So we're trying to clone every one of those groves, and it'll be beautiful. You have basically the entire genetics of a species on a shelf.
There you go.
To where then we can multiply them at our will, and if anything were to happen out in the wild, we're able to make sure that population just can thrive into the future.
So, I mean, it's a really exciting technique that's being applied many levels. We're going to go to questions next in a second, but just to touch really briefly on the ethical considerations. So, obviously, there are there are indigenous communities who are really invested, of course, in the conservation and deeply connected to protecting these plants. So, wondering if we could, as our final question, I'd be interested to hear about how that kind of community is involved in this technique.
Yeah. So that's something we're looking at with, especially on Hawaii and, on Catalina, where we had a long history of the Tongva that went out to Catalina. It was more like Kumie down in the southern islands, and up in the northern islands, the Shumash. And getting them involved in this idea we have. They were stewards of the land for so long before us. And when we are looking to do new methods on how we can save these plants, getting involved on how the right guidance and to do those types of things. And this is really big on Hawaii too, and a very exceptional scientific community that has been eons with indigenous peoples there as well. And when I learned it, I learned from some incredible people that were using and looking at these as tools to be good land stewards and species stewards. Like I said, hey Jeff, that tree, and it's like these are personal. This is personal. These are our neighbors too. So, yeah.
So I think we may have some audience questions. So if you do have a question, if you raise your hand, one of the Natural History Museum staff will come over with a microphone. Just on the left, I think we have a question.
Hi, I'm curious and really fascinated by this stuff you're telling about, but I just keep on thinking about with these very rare trees, the next steps to maintaining biodiversity. Since you're making so many clonal copies of these, how do we maintain, get things like the ironwood to start breeding with the other populations and maintain more biodiversity?
Yeah, so great point. So when we're talking about really low population size, the very first step for rare plant conservation is to ensure that these don't go extinct. So the rush in this is to really make sure that we have effective and reliable methods that we can preserve the genetics with that. And so when you have challenges where the seeds are not really being produced, we have this tool available to us to overcome those challenges. So first step, make sure it doesn't go extinct. We'll get to the other questions later. So in the case of the mountain mahogany, that one, we know that we have this really rare population and it's being surrounded by that hybridization. So our goal is to then clone and capture every genetic tree. We actually just found out that there's been possibly one other that was gifted to the Berkeley Botanic Garden up there. That might be our 7th tree. It was done almost 100-something years ago before all this damage happened, and we just expanded our genetic pool there.
Oh, that's exciting.
So we'll be able to capture all those genetics and then create what we call conservation groves at places like the Natural History Museum, if you guys are, any of the horticulturists are here are interested, where we can create and have every 6 of those individuals here where that other tree is not around, and hopefully we'll start to have pure individuals and seedlings coming up. And so we have partners at the San Diego Zoo, Cal Botanic Garden, which used to be Rancho Santa Ana Botanic Garden out in Claremont, Santa Barbara Botanic Gardens and other places where we work with botanic gardens as other places to safeguard and have conservation of these. And then we can possibly, hopefully, get good, viable, pure seed to that true to that species. And then we can go back to other methods like seed banking as well.
Yeah, and preserving that biodiversity also in the context of invasive species as well, where it's kind of the opposite problem because it's unchecked.
And these plants, and once we have living plants on like I said, a whole, all the genetics of every individual on a shelf, that's also basically essentially exactly what we're doing with seed banking. But you have a living plant. Which a seed is. It's a little baby individual within that plant. So it's a different way, if you can conceptually look at seed banking. We use that term called germplasm storage. So we're storing the germplasm or the genetics, and we have propagules. It's just not a seed; it's a living plant in a tissue culture setting.
Several steps ahead. On the right
Leads
here.
perfectly into my question about
Okay.
seed banking and citizen science. What can we do to help you save some seeds?
Okay,
Or save some plants?
I love this question
It's a great
because
question.
it's not just the seeds, but what I would encourage all of you and everybody here, become a gardener. Learn horticulture. And in your garden, you are essentially doing exactly what we're trying to do at these places that have these conservation groves or anything like that. If we can grow rare plants, and a lot of them are so beautiful and are part of a really functioning system on the islands, in my case on Catalina, but they can also be a very beautiful part of your garden. And in that case, you're actually also that steward that's taking care and ensuring that a rare plant is no longer going extinct. If anything was to happen to that plant out in the wild, it's safely guarded in your garden. And then maybe we can collect seeds as well. And with citizen science, a lot of the gardens that do seed banking, they'll take your seeds too. If you say, hey, I have this really rare plant, hopefully if it's like permitted and all that, that's one thing, but that's between you and whoever issues those permits. But that is essentially, you can be a big part of ensuring that these plants do not go extinct. And you can. So I encourage you. I know a lot of people say, I don't have a green thumb. Well, like anything, it takes practice and a little bit of education. So
And
you
there
can do
are
it.
lots of resources as well. There's the first resource, if you wanted to get into this, if you were inspired this evening, where would people go?
Yeah.
Could go to your website or to
For resources for micropropagation
Yeah.
specifically, yeah, unfortunately, and that was the challenge I had, was there was nowhere really teaching this. You had to it was either a class in a graduate school, but luckily I was able to find one in Hawaii where they were offering us a certificate program, which unfortunately I heard is no longer a thing anymore. But there's books. There's a great book that is out there. It's called Growing Plants in Test Tubes. Fantastic book. It goes through all of the steps. It even gives you the recipes on how to grow a lot of these plants and the why that you're doing it. Back to citizen science, I mean, YouTube and people sharing that information. There's a lot of people out there that have had it, and I've given, I think, a couple. I mean, it's more towards rare type plant conservation, but there's a lot of information out there now. And like I said, there's a couple companies now that have specifically made tissue culture more available to home gardeners. Phytotechnology is one I really like. Phytotech laboratories or something, and they have a pretty relatively cost-effective way for people at home to start using this method as well.
We have another question at the back.
Thank you so much for this educational conversation. My question is, are there, and you may have touched up on this while you were speaking and I may have missed it, but are there external or internal factors or reasons why certain plants like the Catalina Island mountain mahogany don't produce seeds anymore? I think about as a human, right, as a woman, you age and your ovaries start diminishing in the number of eggs they produce. Are there reasons or factors why this happens in plants?
Yeah, so with the mahogany, it's more about a hybridization issue where we don't get pure seed. But the ironwood that I showed there is a great case for that. And a lot of that has to do with the low genetic diversity. So when we get to really small population sizes, weird things start to happen, and sometimes internal, just the inbreeding of certain, or the plants are just a little too closely related. Things will start to happen, and plants will start to look at resources, and they'd be like, okay, they'll start to produce, you'll have signals from the environment where it says it should be producing an embryo or something like that, and it's like, this isn't going to survive, and we have to, and this is biology, We're living beings, and unfortunately, things go wrong, and so flowers and things and embryos will be aborted just naturally. And it'll invest in other strategies. Okay, I have to go and use a basil sprout, and that's how we used to have all those trees that are clonal. It invested in that strategy instead of the seed strategy. So it might be evolutionary or other reasons why, but unfortunately, with the case of the ironwood, a lot of it is we were thinking it's also climate change. That these trees are getting more and more stressed. There may be an extinct pollinator too. We don't know. So that's other things that we as scientists have to try to ask questions and use methods to be able to try to answer those questions.
Yeah, can you just touch on white sage and how much of it is poached, and people that love to burn it, and so much of it is poached from the wild?
Yeah, that's a good question.
Okay, so white sage. Sorry, I missed it at first, but I now got you. So yeah, white sage being poached at the wild. So again, this is methods where this was culturally done for so long, and people have started to pick up on those trends and try to use them themselves. And these plants sometimes when the market suggests and makes it available, and that information got out, and more commonly where people would use saging and smudging for whatever reasons, that once there's a supply, or once there's a demand for it, then it looks to the supply. And if there wasn't enough ethically done or ethically sourced, that's where it can be a challenge to where you're going to supply them. And it's out there in the wild. And white sage only occurs in a certain region, and that's here in our Mediterranean climates here. And so they were poached quite a bit to supply those markets. And again, I encourage you all to become gardeners and grow your own. And that way those plants are it's a beautiful plant. There's so many pollinators that love white sage. Hummingbirds are a great example of that. There's many caterpillars that like to use it as to become butterflies and moths and things like that too. So well beyond just the smudging practices, you can ethically and sustainably grow that in your own garden, and avoid that poaching kind of effort.
Thanks. That was a great question, and I know white sage is being overly harvested, so that's great to know that we can grow our own. Kevin, thank you so much for this discussion. That was so educational and inspiring. It's been such a pleasure to hear about micropropagation from you, and I know that you're a real leader in this field, and we'll be excited to see kind of where this technology goes.